Introduce Dr. Michael Yaffe, Associate Professor of Biology and Biological Engineering at MIT. Dr. Yaffe has been a member of the MIT faculty and the Koch Institute since the year 2000. He received his undergraduate degree in material science and engineering from Cornell University and his MD and PhD in biophysical chemistry from Case Western Reserve University. He completed his residency in general surgery and surgical oncology at Harvard Medical School, and he did his postdoctoral training in the Department of Cell Biology at Harvard. Dr. Yaffe is now an attending surgeon in the Surgical Intensive Care Unit at Beth Israel Deaconess Hospital. He serves on the scientific advisory boards of several biotechnology and pharmaceutical companies, and he is the author of over 130 scientific papers and reviews. An important result of his research efforts is the identification of a defense mechanism that helps tumor cells to survive chemotherapy. Specifically, he has determined that many tumors use specific signaling pathways to survive. Drugs that knock out this mechanism leave tumors much more susceptible to chemotherapy, opening the door for lower doses of chemotherapy with fewer side effects in patients. Please welcome Dr. Michael Yaffe.
Thank you very much. I know you guys have had a long 2 days, so I will try to keep my comments relatively brief. I also know I'm talking to a very sophisticated audience, so I will at times delve into things relatively deeply, but I'll try to move from one area to the other with enough versatility that everybody, hopefully, I can touch on areas that are of interest to everyone. I should say, as Sally mentioned, I wear 3 hats, so I'll jump back and forth as best I can between the hat I wear as a biologist, the hat I wear as an engineer, and the hat that I wear as a clinician. And I'll start by showing you something I'm sure you've seen before. And this is Bob Weinberg's wiring diagram of a cancer cell. And this immediately leaves us with the problem that I know has been enunciated by several of you already, which is if there's so many things going on within a cancer cell, how do we pick which ones to study, and what if we happen to pick the wrong ones? And so I think there are really 3 or 4 key questions that we should be asking ourselves in trying to understand the causes of cancer and the way in which we can treat it. And the first one is, what is it about cancer cells that's different from normal cells? What is it about how cancer cells process information and make decisions to grow or not grow or to metastasize or to grow without restraint that's different about a cancer cell and makes it unique compared to a normal cell? The second question is, with all of these pathways, do cancer cells care about only one pathway or only 2 pathways, or do they somehow read the information from all the pathways and integrate it in some complicated way to make the decisions about whether to grow or not? And finally, how does cancer function as a tissue? Maybe we shouldn't be doing the things that our lab and most other labs have been doing for years, which is to study the houses, if you will, the cells. Maybe we need to think about the whole city. Okay. Finally, can we use the knowledge? If we had a magic way of being able to read not one pathway or another pathway, but many pathways at the same time, how could we use this knowledge to predict a patient's prognosis and maybe improve the methods of treatment? That is personalized cancer treatment the right kind of an approach? And the way that we've been pursuing this here at MIT, as I know you've heard for the last 2 days, is by trying to build an interface between engineering and biology that's centered on cancer research. Now, of course, this is, you might say, well, this is in essence a merger of not just 2 different approaches, but 2 completely different philosophies of science. You could argue it's a merger of two cultures, if you will. The biology culture, where we try to reduce everything to the most simplified elements, and the engineering approach, where we actually try to take things in their complicated state and understand them and optimize them. And so if we break this down, we could say that what we try to do as biologists is to define a problem within some general scientific context. And historically, the way we've done this is by picking one molecule, P53, P21, RAS, or whatever, and study that molecule to death. Put one postdoc or one student on it after another. And then hopefully use things in which we can manipulate genes as proteins as the relevant objects. And this, of course, works quite well to a point. But it's not very good if you're trying to study a complex system like a cancer cell. The engineering approach, and I used to work as an engineer. I worked briefly for the Navy. I worked for Hewlett-Packard. So I have a little bit of practical experience, which is quite different than necessarily what we got when we were trained in engineering. What I learned from there was that the engineering approach really involves using multiple levels of abstraction. Maybe the relevant object shouldn't be a gene or a protein, but should be something else. Maybe it's not even a physical construct. Maybe it's some mathematical synthesis of genes and proteins. The idea behind engineering, of course, is to make measurements, to build and design tools for measurements, and to use those measurements in a very quantitative way in order to be able to use sort of continuous or high throughput monitoring of a system or a circuit or a chemical plant to understand what's going on, and to be able to build predictive models even with incomplete data. And I can tell you as a clinician, most of the time when it comes to treating patients, this is what we have. We very rarely have complete understanding of what's going on. We often have to make very important clinical decisions with very incomplete data. And so I think the merger of the biological approach, where we can think in depth about one process or one molecule, with the engineering approach, where we try to fit that into a broader context, has real potential to advance our ability to understand and treat this terrible disease. So one way that I like to introduce this to lay audiences that aren't as sophisticated as you are is to say we should think about this, for example, as a house. And what a typical biologist might do, for example, is focus on the plumbing system, and he might focus on the bathtub. He might devote his whole laboratory career to trying to study that one bathtub in this one plumbing system. And another biologist would be equally interested perhaps in studying, say, the thermostat as part of the heating system within the house. And another biologist might be equally interested in studying, for example, the blower that sits in your attic that helps to run your air conditioner. And it's like the blind man and the elephant. Everybody's feeling one thing and thinks they understand what cancer is all about. What we do as engineers, of course, is we can't think that way. We have to think about the plumbing system in the context of the heating system, in the context of the air conditioning system. So we have to keep in mind all of these complicated things that are all happening at once. Another relevant analogy, for example, one that's going to get us one step closer from that abstract thing to what it is we actually do in the lab, is perhaps we should be thinking about this like it's an electrical circuit. After all, that's really what a cell probably is best considered, a series of electrical circuits in which what we really want to do is understand how information flows through this electrical circuit. Where are the currents and voltages that are of interest? What are the inputs and what are the outputs? So how does signaling actually work? How does it control inputs and outputs? And we could take any circuit that we wanted to, in this case it's a microprocessor, and we could define what the inputs were. A voltage comes in here, another voltage comes in here, there's a reset switch set over here, and another input voltage is set here. And as a result of that, the system, the circuit, gives us a certain output. It gives us an output through this VGA port. It does something to the speaker. And interestingly, the same port in which we put an input voltage actually also can deliver an output voltage. So we can define inputs and outputs. And what we really want to know, if we're going to treat a cell like an integrated circuit, is what are the currents? What are the voltages? But clearly, we can't measure all of them. Now, what we know as engineers is you don't have to measure all of them. You just have to measure the right ones. So the key is to pick certain test points in the circuit where the voltages are critical predictors of what the output would be if we knew what the input was. So we don't actually have to measure everything. We just have to somehow figure out what the key nodes are that are going to be the test point for our circuit. Okay. That is, maybe in a biological circuit, if we had a meter that could measure the activity of certain proteins, a kinase called JNK, or a protease called caspase-8, maybe we could predict if a cell would live or die, or if it would grow if we stimulated it with a growth factor, or if it would metabolize glucose differently if it was stimulated with insulin, by measuring not the activity of all of these proteins that we overwhelm each other with by showing these complicated diagrams, but only a few things. If we just had accurate enough meters that could measure the relative currents and voltages in a biological system. Now, the electrical analogy of this, of course, is just a probe card. You take your integrated circuit or your breadboarded circuit, and you plunk it down here in the center of this test probe, of this probe card, and you measure the relevant, it's like a bed of nails tester, and you measure the relevant voltages at a dozen or 2 dozen or maybe even 100 points, and that tells you all you need to know to understand what's going on with your circuit. And that's what we need to do here. We don't need to measure everything here. We can't possibly do it. We only perhaps need to measure a few things: MAP kinase activity, or RAC activity, or the activity of a kinase called AKT, or a transcription factor like NF-kappa B that Tyler talked about. And so what we want to figure out is which nodes are carrying the critical information, and what information is it that they're carrying. Now, in order to do that, we have to describe what we mean by information. What constitutes information? And Tyler gave you one example of that. That's gene expression. And we often run around and measure the expression of a lot of genes. But you know, the downside of that is it has been, in my very, very biased opinion, it's been a combination of spectacularly successful in what we can measure and spectacularly unsuccessful in furthering our understanding of what it is we're measuring. And that's because when you measure the increased expression of a gene, that doesn't mean that anything's happening. That's just the potential of the cell to do something. You know, in the movie Scrooge, he looks into the future and he says, is this the way things are, or is this just the image of the way things might be? And when you look at gene expression, you're looking at the image of the way things might be if those genes were actually turned into proteins and those proteins actually worked. Well, that's not going to work if we want to measure an electrical circuit. I don't want to know what the circuit might be able to do under some condition. I want to know what it's doing right now. And that's a lot like the game that we used to play as children called Pass the Red Ball. We used to play this when I was a kid. You pass the red ball along, somebody plays some music. And when the music stops, who's ever got the red ball, they're it. They're the winner or the loser. They're the person who's holding the information content. And in cells, it turns out that the red ball is a phosphate group. And the way you can tell for many proteins that they're active is if that red ball is sitting on the protein, if that protein contains a phosphate group in the right place, it's active. So the way we're going to measure currents and voltages in our cells, instead of measuring gene expression, for example, is we're going to ask what proteins are phosphorylated. And if they're phosphorylated, that must mean they're active. That must mean that information is flowing through that circuit. Okay. You know, let me go through. So one way that we can find out who's holding the red ball is by quantitatively measuring things using mass spectrometry. We could take a cancer cell, for example, and we could treat it with a chemotherapeutic drug. And this work that I'm going to talk about is all done by Forrest White's lab, a good collaborator of mine. And we could break the cell apart, and we could ask what proteins are phosphorylated at various points in time by using a very clever trick, using mass spectrometry and chemical modification. We take all the proteins that are phosphorylated from samples that are treated with a DNA-damaging drug, say at 1 minute, and 5 minutes, and 10 minutes, and an hour, and we label each one with a compound that contains a slightly different mass because we use compounds with slightly different isotopes. And then we can do mass spectrometry and just look at the weights of the peptides that we get back, and that tells us not only who's phosphorylated, but how does that phosphorylation change in time. That's like not just asking, well, what's the voltage at those test points, but what's the voltage in the circuit at those test points as that circuit is actually doing something, like growing or responding to chemotherapy. And one of the things that this has been used for very successfully by Forrest's lab is to look at the reaction of cells to treatment with inhibitors of the EGF receptor. So the EGF receptor, as I suspect many of you know, is a growth factor receptor that seems to be associated with a wide variety of lung cancers. Lung cancer, breast cancer, prostate and colon cancer in particular, and now some emerging data over the last 10 years that head and neck cancer might be particularly responsive to the EGF receptor. And the reason it's important is because there are drugs that are out there, antibodies that block the function of this and small molecules that block the function, that are actively out there in clinical trials. And so clearly it would be useful to know what is the signal that the EGF receptor is sending and how do these drugs work? Who should get these drugs? How do we figure out what those drugs are doing? Now, it gets a little more complicated, as all of biology usually does, because unfortunately there's not just one EGF receptor and there's not just one EGF, but there are in fact multiple types of EGF and at least 4 different types of receptors, and they send their signals through these complicated pathways that involve many of those nodes. But by mass spectrometry, we can measure in real time who's phosphorylated, which protein's carrying the red ball, where is the current and voltage going in this circuit. And what I'm showing you here for each protein is a different site on the protein that's carrying that phosphate group. And I'm showing you what happens in 5, 10, or 30 minutes of treatment, where things that are red mean they're very highly phosphorylated, and things that are yellow are less so, and things that are green are less so. So now we can measure. It's like that bed of nails tester. We can measure how information flows through this circuit. We've been able to identify from that a couple of the test points that we think are important. And as a result of that, out of that big complicated network, if we look not just at who's phosphorylated, not just at what the voltages are, but where do the voltages change, we can break the entire behavior in this case of breast cancer cells, as far as cell migration goes, or cell proliferation, into only having to measure 5 things. This greatly reduces what we need to measure. We only need to measure these 5 proteins in order to be able to get a readout, to be able to read the meter for whether that cell is going to proliferate or migrate. And this is obviously of great importance, for instance, to drug companies that want to develop new drugs. Can we uncover anything else with this? Well, what if instead of playing around with cells in a culture dish, something that my lab does a lot of, because we have only recently begun to become as sophisticated as Tyler's lab using mouse models, in part because we've partnered with Tyler's lab to do it, but we can do this with mass spectrometry with real tissues. And in this case, the work that Forrest Lab has done is using tissues, human tissues, from a disease called glioblastoma, a disease that perhaps wasn't quite as widely known until Ted Kennedy had the great misfortune to come down with this. So this is a terrible brain tumor. It's a brain tumor that in some people starts as a primary tumor, and in others it starts as a lower grade tumor that over the years becomes this terrible tumor. And the tumor is characterized by rapid proliferation, diffuse invasion, growth of new blood vessels, and ultimately pretty rapid death of a patient who has such a tumor. And many genetic alterations are known to occur in these tumors, but one of the most important ones was mutation of that EGF receptor that I mentioned to you earlier. A subset of patients turn out to have not just that EGF receptor, but a mutation called V3. And this V3 containing glioblastomas are the worst kind, because patients who have mutations in the EGFR, this V3 mutation in glioblastoma, are generally dead within 4 to 6 months after diagnosis. So we would really like to learn some way that we could particularly treat these patients. And so using genetic tools of the type that Tyler mentioned, we've been able to look at tumor cells that express either relatively modest or even higher levels of that mutant protein, or a form of that mutant protein that doesn't work, and ask what's different in these tumor cells. And what came out of that was the observation that there were only a few pathways that were different in the tumor cells that had a terrible prognosis and the tumor cells that had a relatively reasonable, I guess reasonable is not the best word, had a better prognosis. And as a result of that, it turned out that we discovered 2 things. The first was certain sites on the EGF receptor were important. And the second was there was a pathway that we hadn't even been looking for that came up out of the mass spectroscopy. And that was something that went through a different protein called MET. So what this was like, for example, was it's as though what we had set out to do was to look at the EGF receptor. We thought we were studying the plumbing system. But because we did this systems biology approach, we found out the real problem wasn't necessarily just the plumbing. It was something about the plumbing and the heating system together that was the problem with these tumors. Now, as I said, these tumors are dramatically resistant to therapy. If we take those tumor cells and we treat them with cisplatin, a very common chemotherapeutic drug, you can see that compared to the untreated cells, we can't kill these cells with cisplatin at all. This is survival. There's barely any difference in the survival if we treat these cells with cisplatin. But as I said, we discovered from that systems approach that there were 2 pathways that were important. The EGF pathway, that's the plumbing we thought we were studying, and this MET pathway, that's like the heating system that came out unexpectedly. And what we found was that if we took those cells with the same dose of cisplatin at which they were completely resistant, and now we inhibited either of those 2 pathways with drugs that are almost, but not yet, in advanced clinical trials, we could make those tumor cells remarkably sensitive to chemotherapy. And so what this suggests is that if we take this systems approach to studying tumors, we may be able to find new treatments for tumors that we've always thought of as being recalcitrant. Okay. Let me spend the last part of the slide giving you one more example of this. And that's a subject that my lab works on in significant depth, and that has to do with the DNA damage response. Now, Tyler mentioned, and I know you heard from Alice and several people yesterday, that one of the hallmarks of cancer is the fact that cancer cells evolve because they contain mutations in their DNA. They contain damage to their DNA. But in fact, the very thing that caused the cancer, the mutations in the DNA, is also the Achilles heel of the tumor cell. Because when we treat patients with chemotherapy or we treat patients with radiotherapy, those work because we're damaging DNA. In fact, as a number of my colleagues like to say, all we do as oncologists really and as oncological surgeons is barbaric. We either try to cut something out with a knife or we feed our patients poisons. That's all we really know how to do. And most of the poisons that we give patients work by damaging DNA. Not all of them. But it would be nice to know when we should give these poisons and when we should give the other kind of poisons. Now, as I said, they work by damaging DNA. This is cisplatinum. And this is an example of a drug that works because it binds to DNA and damages it. Now, most cells, any self-respecting cell that has normal genes, has a normal p53 gene, has a normal p21 gene, when it sees damaged DNA, it hits the brakes. It says, stop. I'm not going to try to divide. I'm not going to try to do anything till I fix my DNA. But a cancer cell is different. Its wiring diagram is different. That's how it became a cancer in the first place. But it also means that that cancer cell can't stop. And even though it's got broken DNA, it's still barreling along trying to divide. That's like thinking of a cancer cell as an automobile that's going at 150 miles an hour. And now if you throw something up in front of it, well, it can't hit the brakes. There are no brakes. And as a result, it crashes, and that's the end of the tumor. So if we could figure out how to make DNA-damaging chemotherapy more effective, we'd be in great shape. And again, this is an example of how we've used a systems approach. So what we've done here, this is one of those wiring diagrams of the cancer cell. And what we've done historically over the years, as have many labs, is to pick one part of this complicated pathway and maybe pick one protein and just beat it to death. And I'm here to tell you that that approach is about at the end of its useful lifetime. And so in the last few years, what a number of people in the lab have done, Kevin Janes, a graduate student, Andrea Tentner, a graduate student, Jerry Ostheimer, and Alex Gardino, 2 postdocs, is to begin to explore not a single protein or a pathway, but a whole variety of proteins and pathways all at once, by measuring their activities, their phosphorylation, if you will, not just of a single protein, but of all the proteins that you see highlighted or marked here. Now, of course, and I'll skip this. This is just how we've done it. I'll come to that in a minute. So what they've done is take cancer cells, damage their DNA, and then measure about 40 proteins or so at all these different times shown here. And then they measured what the cells did wherever you see these red and blue balls. So they're looking at whether the cells divided or whether they died. And what comes out of this, of course, are these very complicated trajectories where you're looking at each one of those different molecules that might be important, and you're looking at its activity as a function of time. Okay? Now, what you end up with here is something which to a biologist is a nightmare, but to an engineer is something we're very familiar with. A very complicated data set that tells us about all the measurements we've made and all the responses we've made, and no obvious way to relate hundreds of variables on one hand to a small number of observations on the other. But as engineers, we're all used to this because this has been our bread and butter forever. And we know that there are a bunch of computational methods of dimensional reduction that will let us do this. So as a result, what we've done is to take and modify some methods that we think about as engineers and apply them now to biology. Things like principal components analysis and stepwise regression. 3 things have come out of this, and if I have time, I'll tell you about one of them for about 5 minutes. One of the things that came out of it was new pathways that were important for DNA damage responses. A pathway that I'm going to show you if we can inhibit clinically, I think we're going to really make a big difference in being able to treat a subset of patients with personalized chemotherapy. The second thing that came out of it was something that I found so surprising, it still boggles my mind. And that was, I guess I had always thought that a component in a cell did one thing. After all, a resistor in a circuit is a resistor. It doesn't behave like a capacitor one day and an inductor another day and a resistor the third day. But that's not true in biology. In fact, a component, a molecule in the cell, under one conditions can send one signal. It can tell the cell to survive and divide. And the very same protein, under different conditions, say when we treat cells with DNA-damaging chemotherapy, can say the exact opposite. It can tell the cell to stop growing and die. And the third thing, which I won't have time to talk about, but my colleague, Michael Heeman, who will speak after me and with whom we've done this work, will tell you about how we can use these 2 observations to understand something called synthetic lethality, to find a way that if we could personally understand every patient's tumor, and I think we can, we would be able to design customized therapies that would work for every patient's tumor in a unique way. Okay. So let me simply get to the punchline here and tell you that what emerged from all those complicated measurements and the dimensional reduction was the finding that there was a pathway that nobody had ever expected to be involved in how tumor cells survive after DNA damage. Everything over here was known. In fact, our lab had helped, along with a number of other labs, to determine this part. What we found that was so surprising was this bit. There was another pathway that nobody had been aware of as being important for chemotherapy. And the reason is, this is a pathway that's generally thought to be involved in how cells respond to all kinds of stress. It's how a cell responds to low glucose or to low oxygen or to high salt concentrations. But nobody had thought that this had, including us, had anything to do with DNA damage, even though this pathway had been known for many years. And it all involved one particular protein called MK2. Basically, what we found was that MK2 was required for tumor cells to survive chemotherapy. So this is a mouse, one of those mouse models that Tyler talked about. And what we've done here is to inject tumor cells in the flanks of the mice and let the tumors grow. So here's the live mouse. Here are the tumors, which stain green under this kind of imaging. And here's what they look like. And these are just control tumors. And the only difference between the tumors on the left and the tumors on the right is the tumors on the left are tumors in which we've experimentally knocked out, knocked down, gotten rid of that molecule that I told you our systems approach had revealed as being important. And you can see that when we do that, oh, it's terrible. The tumors, if anything, are bigger under controlled conditions than the tumors that have that protein. But now if we treat those mice with chemotherapy, conventional chemotherapy, cisplatin or doxorubicin, at a dose that's so low that it would have no effect on the normal tumors, you can see it makes those tumors that we've gotten rid of our protein in basically melt away. And so what it means is if we can target this protein in tumor cells, we can make those tumor cells sensitive not to some fancy new drug that has to be invented, but to drugs that we've had in the clinic for the last 30 years. The most interesting thing is it only works. It's only effective in making cells sensitive to chemotherapy if they're missing p53. So all of these bars are the same, except for these last two, the gray one and the white one. And the difference here, and what I'm showing you, is how much those cells survive as we increase the dose of chemotherapy. And these, the ones that are preferentially dying, are ones where we've knocked out our molecule, MK2, and they also don't have p53. Why is that important? Well, it's important because a lot of tumors don't have p53, but all of the normal cells in your body have p53. This means if I come up with a drug against this molecule we found, it's not going to make your normal cells any more sensitive to chemotherapy. It's only going to make the tumor cells that don't have p53 more sensitive to chemotherapy. It's as though we could custom design a therapy that would only work against tumor cells. And it'll work with the same old chemotherapy that we've had around since before I went to medical school. And I assure you I'm a lot older than I look. So what does it mean? This, I think, will be the last slide. What it means is that normal cells get by after DNA damage, and this represents different types of DNA damage, because they have two pathways, and they need both pathways in order to survive. Cancer cells are missing this pathway, and as a result, they cheat, and they take another pathway, a pathway that ordinarily is not important, and they make it essential for them to survive DNA damage. What they've done is to rewire their circuitry so they have an AND gate. So they use this pathway that all cells have, plus this new pathway that all cells have, but only these p53 deficient cancer cells require. And in fact, a drug that's been through clinical trials and is already out there, we stumbled onto because, in fact, it inhibits both branches of this AND gate. And that means we can potentially take a drug that's already been through some clinical trials and been given to patients and use this drug together with conventional chemotherapy to sensitize tumors in the case where the tumor cell is missing p53. Let me just get to the last slide because I think I'm about to run over. Let me show this one more slide and then I'll move on. Okay. So this leads to the concept of synthetic lethality. And I think Mike Heeman, who follows me, will tell you about it. The idea that maybe we could use DNA repair pathways, that is, we could target those molecules I told you about as targets for cancer therapy if we did this in a clever way. That is, if we made use of the fact that we can sensitize tumors or predict patient responses to chemotherapy or radiation, if we understand what the circuitry is and how it's miswired in tumor cells. Now just to cut to the very last slide. Okay. So what's the future going to hold? Well, I think on the horizon there are a couple things. In addition to the systems approach that I've told you about, one thing that we would really like to understand is what's the instruction set for becoming a cancer cell. If you think about the genetic code, not like a biologist, but like a digital engineer, you would think of it as being zeros and ones. And what's different about a cancer cell is some of the things, for example, that were zeros in a normal cell are likely to become active in a cancer cell. That would be turning on an oncogene. And some things, for instance, that were active in a normal cell are shut off in a cancer cell. That's a tumor suppressor gene. And so if we can think about what the code is that converts a normal cell to a cancer cell, that'll help. Particularly if we think about the code, not in general, but the code for every patient's unique cancer. The second thing, of course, is everything that we've done to date has been to study cancer at the level of individual cells. But that cancer isn't just a disease of houses. It's a disruption of a whole city. And this is, I think, the point that Tyler made, is sometimes we can't just look at the cancer cells. To really go the next step, we have to look at the whole city, the cancer cells and the world in which those cancer cells live. And then this truly is my last slide. I just want to say that I think where we're stuck right now and where the future is going to take us with this combined engineering and science approach is further along the line of increasing the number of components that we can study in mechanistic detail. At the moment, we're probably somewhere over here, where we can study a lot of components. But the more components we study, the less detail that we understand them in. And as a result, there's a compromise in trying to score predictive power based on the things you can study and the amount of detail you can study them in. The hope, as we move forward with our engineering approaches, is that we'll move along this curve here so that our ability to understand a lot of components in mechanistic detail moves from this solid line to this dotted line. I'll stop there, and I'll thank you very much for your attention.
How about that compound that you came across, Mike? Where is it standing?
So you can. It's been used in clinical trials. Now, most of the trials that it's been used in have looked at it only as a standalone compound. And that's because until our paper came out a few years ago, no one had thought about trying it together with chemotherapy. It has been tried together with chemotherapy since then in a subset of women with triple-negative breast cancer. Very small number, because it's only used on compassionate basis. And in 9 patients who received it, my understanding is that 4 of those patients have had complete remissions. So hopefully, that drug or something like that will move forward. Together with Tyler's lab, a former postdoc in my lab, who's now set up his own lab in Germany and an ongoing postdoc, has now made a mouse in which we can specifically give a drug that targets that one particular kinase. And I hope to win the $10,000 from Tyler, because we're breeding that mouse against the lung cancer mouse he's got, and I hope I can cure the cancer in that mouse. I think we've at least got a shot at it.